Hi, everyone. Thank you for joining this week's Single Cell and Spatial Exploration Club. For this session, and actually I'll share my screen so you're able to see that a bit clearer. So I'm glad everyone is joining today to get a bit more information on some of our new updates regarding the BioVinci platform, as well as a new case study within the BBrowserX Pro. So I'm glad to see everyone here. So for today's session, as I mentioned, this session will focus on some new updates within the Vinci, now sort of termed BioVinci, on the BioTuring ecosystem, which is our main platform for centralizing any specific visualization, whether that's chart editing, sharing data across teams, or being able to export the data in sort of high quality formats for publication purposes. So that's going to be one of our main key things is really going through the process of sort of doing that in the system, as well as how can you actually share projects across different sort of colleagues in your team, and really looking at how are we able to apply a sort of case study. Let's say for this week, we're focusing on functionally mapping glial and neuronal specific cell populations in neurodegenerative diseases, and really trying to sort of visualize these sort of questions across different types of tools. So that's going to be sort of the focus of today. So just as a recap, so BioVinci can now be found within the ecosystem. So you're now able to directly access the beta version of BioVinci. We will be sort of working on in future versions, once this is sort of a system that is fully released, we'll be sort of supporting direct data submission. So that's going to be something that you can also work on within BioVinci, being able to upload the data that you're working on in the system and being able to create your own visualizations from that data. And also expanding other utilities within BioVinci, for example, visualizing and running other statistical analysis as well. So that's going to be sort of in the future roadmap, which I'll also show how you're able to find that within the system. So if you're interested in that, please feel free to try the new BioVinci and really sort of the features that you can sort of apply. So another thing that I'll come back to, so now if we go into how BioVinci will be centralized within the ecosystem, so how are we able to connect and generate our own visualizations and export them from these different sort of platforms into this new system. So you'll normally see that the BioVinci icon will be sort of connected within SpatialX, which will be available very soon. Currently, you're able to use it for SmartBulk as well as BBrowserX Pro. And some functionalities of talk to data. For example, if you're trying to analyze a public data set from the repository and you're exploring that study in the system, then it'll sort of connect that public data set into BBrowserX Pro. So that's where you'll be able to sort of generate different types of visualizations from basic charts when you're trying to look at proportions or the level of expression of a particular gene across different cell types or different conditions, to what we would see in sort of the SmartBulk application, which is like deconvolution of different cell type proportions across the data within that system. So these are some of the ones that I'll sort of try to cover today within BBrowserX Pro. So the data set that we'll be using today as an example sort of case study and sort of the background disease for this specific webinar session will be this new data set that has been added into the system, which is really looking at how are we able to look at different subtypes of glial cells that might contribute to Alzheimer's disease or Parkinson's disease specific neurodegeneration. And particularly other populations, not only sort of the microglial cells, but also astrocyte specific subpopulations, as well as some other substantial heterogeneous sort of MSN populations within neurodegeneration. So this is sort of a paper that you can find within the Talk to Data system. So if you're interested in knowing how you're able to do that, you can access the new Talk to Data version 5. which is the human database of the system. You can query specific genes. So if that's something that you're interested in, is sort of identifying what studies in the database are expressing or have cells that are expressing a particular target, you can enter that specific target in the system, and you can identify what specific data sets particularly are showing the expression of this gene, as well as the specific threshold expression of that gene target or gene marker. So that's one way you can search through the database, and you can specifically add the disease models that you're focusing on. So are you looking at, let's say, APOE within a specific condition? That's where you can add what these conditions are. So you're able to create that ontology map relating to that disease or that tissue. Or are you specifically looking for a specific threshold of that gene expression across the database? So do you want to see, okay, what is the highest sort of average rank threshold? In what cell type populations do I see that threshold, and what studies is that information coming from? So you're able to get an idea, is this tissue specific? Is this disease specific? Or even is this a gene that would be sort of specific for like treatment back? So this is just one way. The other way, if you just want to see where that study is in the system, you can also search for the study name. And that's where you would be able to explore that individual data set, which will be migrated into the BBrowserX Pro for further downstream analysis. So that's just a recap, so you kind of know where you can find these features and how you can access them in the system. So if I go into sort of the case study introduction and a bit more on why we are focusing on neurodegenerative diseases, it's because Alzheimer's disease and Parkinson's disease really impose a growing global burden. And even though we have identified specific cell populations that can be targeted or have specific mechanisms that might drive severity or sort of further neurodegenerative symptoms, we're still not able to really understand the divergent pathologies within these cell types specifically, and how these mechanisms might sort of change these pathologies further. So there are a lot of studies that have been published recently, which are available on the Talk to Data system. One, which is the Shu paper, that is the one that I'll go into more in detail, which is this one right here, which really tried to look at sort of single nuclei RNA-seq data across the human sort of striatal tissue, which is the putamen region. And it really tried to look at what are the differences within these different cell populations when you're comparing Alzheimer's disease, Parkinson's disease to healthy control donors. So really, how can we look at subpopulations and see if they're within activated states, or are they cell types that might be sort of changing specific signatures and might sort of diverge into different transcriptomic differences, or are we looking at sort of heterogeneity across specific subpopulations? So that's sort of the case study we'll go through. And some of the supported, I guess, when we're trying to understand how BioVinci plays a role or can support you when you're trying to do this analysis. So now that it's sort of connected to BBrowserX Pro, you should be able to now export like the TCD and UMAP figure, where you can actually overlay the cell type annotations. That's something that many users have been requesting in the past months. So this is a new feature that you can now sort of run in the system, and I'll cover how you're able to do that from the study. And there are several other sort of basic and functional visualization plots that you can also generate from the data set or the case study that you're trying to run. That includes the blocks plot, which would give you sort of other sort of statistical information, violin plot, where you can look at this sample variation across the plot, and also bubble heat maps, which is more related to the distribution, but also the proportion of cells that are expressing a particular gene signature across clusters or across condition or samples. So that's another sort of figure that you can also generate. So if we go into the study specifically that we're trying to analyze, Let's say one of our first questions that we're trying to do, and I'll go into the slide now for that. So one of the first questions that we'll try to really look at is sort of how can we identify density layers of activated cell populations. Let's say in this case, we're focusing on astrocyte specific cells, and how do they increase or decrease when you're looking at different samples or different conditions compared to healthy controls. So this is a new feature within the system as well that many users were trying to sort of run and they were requesting, which is how can we look at not only the gene expression within the UMAP, but actually looking at density layers within the UMAP. So a bit more on what that actually means. So when we're running density query within the system, what it tries to do, it tries to overlay sort of a density layer in the UMAP or T-SNE embedding to visualize how these cells are distributed within a low dimensional space. And it tries to really look at each point on the UMAP for each cell and sort of create this density layer that mainly gives you sort of the local concentration of that particular gene in the UMAP by using either the kernel density estimation or point density sort of score. So that's a feature where, let's say we want to look at specific expression of particular genes, let's say related to neuroinflammation. So if we go into our first question, like how can we look at these activated states within these astrocyte populations? So if we enter these specific neuroinflammatory genes, and let's say we're querying one gene at a time, let's say we're interested in seeing, okay, how this particular gene target VIM is expressed within these different populations in the study, and can I actually run this density layer calculation? So now instead of only visualizing the gene expression, you're now able to run this density layer sort of analysis. So when you run the density, it should be able to give you that sort of distribution and that concentration score within the UMAP embedding. So we can see that it's mainly focused within the astrocyte population. Now, if we want to overlay, like let's say we want to look at, okay, is this specific to subpopulations of these astrocytes? We can actually rerun the VIM to see, okay, where it's concentrated within the UMAP, and we can see that it's mainly within this astrocyte 1 population here. So that's where you can run the density score. If you want to export this, or if you want to visualize or overlay other information, like how does this density layer sort of differ across different conditions or different clinical stages, what you can do is select the metadata layer for that specific sample. You can then search for VIM or the density score, and you can actually turn off so you're able to see is it different across different populations. So am I seeing different density scores in Alzheimer's versus normal healthy controls? So I would be able to turn off those samples. Or is this different from Parkinson's disease, for example? So that's something that you can do to just visualize first, like these differences. Other ways that you can actually look at these specific scores is if you actually save or label a specific density thresholds, you're able to create an annotation based on that density score for that gene. And that way you're able to actually visualize it either as a violin plot or as a sort of a, not a bubble heat map, but also looking at differences in summary. So how do these scores differ, or the proportions of these scores across the conditions? So that's one way you can start sort of running this type of analysis within the system to identify these differences across different astrocyte populations in this case. So if anyone has any questions, feel free to add them in the chat and I'll try to cover them at the end of the session as well. Okay, so if I go back to the next question, now that we were able to really look at these density features and overlay the density score within the UMAP based on the specific gene targets that we were trying to look at, the next step is how can we actually look at AUCell enrichment within the system, and can we export that outside of sort of BBrowserX into Vinci for further sort of publication editing of the figure. So for AUCell enrichment, I'll use a similar data set because it will need a specific layer when you're running the study. So just in case you're running it on your own or you're trying to explore it on any public data set, Just make sure when you're inputting the genes, it'll normally ask you that the data set will have to have a specific layer of the data. So you'll normally see that if you want to run AUCell enrichment, it will ask you that it needs sort of a specific layer within the study. So it'll need the BBrowserX layer to be able to run that feature. So when you're running AUCell enrichment, what it tries to do is look at how there's specific cells within your study that are active or have a specific enrichment score across the entire gene set. So it's not matching or it's not counting the specific log normalized score for that individual gene, but it's showing you an enrichment score of the entire gene set. So it's sort of a normalized enrichment score. And that's in order to identify whether there's sort of an enrichment of a particular pathway. So if you're looking at specific neuroinflammation genes, for example, these here, you're trying to map out, okay, what cells have an enrichment across the entire gene set, and can I subset these specific cells to look at the proportion across either cell types or how it's driving differences in condition. So that's the sort of reason why you would use that feature. It's whether you're trying to map out an enrichment or you're trying to identify cell subtype populations that would require more than one gene to identify that cell type. So there might be cases for like Th2 cells that might need the expression of not just one gene, but you might need to run very specific markers and an AUCell enrichment to see if you have that population within your data set. So let's say if we want to look at, okay, what cells have an enrichment of these neuroinflammatory genes, we can search for all of the genes within the UMAP, and then this is where you'll be able to switch from match count unit into the AUCell enrichment score. So if we look at the AUCell score, this is where it's going to give a specific threshold. Normally for AUCell scores, we would want to see that there's a sort of clear distribution. So we can see that there clear distribution, that there are cells that have a higher enrichment of these neuroinflammatory gene targets. And we can select a threshold that would sort of mean that that would be sort of a high threshold enrichment score. So that would be normally like 0.5 or higher. Though that would be something that we would want to see within the sort of distribution map and something that we can annotate and label. So if you want to save your AUCell score and not have to run the genes every single time, make sure that you save every AUCell score into your metadata so you know what genes or what AUCell score was run for that specific gene set or that study. So let's say I have my neuroinflammation AUCell score threshold and I want to be able to see that in the metadata. I can then create a label with a specific sort of score threshold. So let's say I want to look at only cells that are higher than 0.5, and I want to create a new annotation. So if we want to add this to an existing group, let's say you have different cell types, but you now want to add a specific sort of annotation for this subpopulation that has a higher enrichment, you can add it or append that new annotation into an existing group, or you can create a new group so that you're able to add all your AUCell scores from different pathways or different enriched sub gene sets. So that's something that you can add, and you can add the layer or the enrichment score that you're adding for these neuroinflammatory genes, for example. Okay, so when you add the new annotation and you create it, that's where you're able to go into your metadata, so private metadata that you're generating within the specific study. And you can actually, once you've created these metadatas from those AUCell scores that are numerical values, it creates sort of a different sort of metadata value that you're able to look at proportions across other either groups or other cell type sort of differences. So if in this case, if we want to see, let's say, differences or distribution differences of these sort of genes that are activated, and I want to see these proportional shifts within different cell types, or what sort of pathway genes will be driving different cell type proportions. This is where we can go into the visualization sort of plots within the BBrowserX system, and you'll have a way where you can generate cell proportions. So now that we are able to see our different sort of AUCell scores for either transcription factors, oxidative stress regulator genes, and these neuroinflammatory specific genes, I'm able to see, okay, how do these specific target pathways have a sort of distribution change across different cell type populations. So I can see, for example, in the neuroinflammatory specific pathway genes, it seems that it's actually within the astrocyte cell type. So it's actually very specific and driving these higher proportions in the astrocyte population as opposed to other populations within the system. Whereas if we look at oxidative stress regulators, it seems that there's a higher proportion of this gene set enrichment within the microglia population. So that's a way that you can start getting a bit of more information from your own gene set enrichment, and you can see these proportion differences. You can layer other information. So if you want to see other information relating to differences in samples, you can also overlay that information and see if it's consistent with just one patient or is it consistent across different patients as well. So this is where you can add that information. So now if we want to be able to see, okay, how can we export this into the BioVinci so I can actually edit it or add sort of a legend into that, there is going to be this connection icon for BioVinci. So you'll normally see that for every platform, and it will be available in SpatialX as well. So when we're exporting these figures into the system, it'll sort of show you once it's imported into BioVinci, and this is where we're able to now sort of adjust the figure so we can add a better legend title. We can add information as well into this. So you'll be able to edit the figures a bit further. You'll be able to change the sort of title of sort of the figure. You'll be able to expand all the different sort of I guess the adjustments or parameters that you can run on that specific chart or the plot that you're exporting into the BioVinci system, or if you need to change the color scale. So sometimes there's specific colors from BBrowserX that might be hard to actually visualize. Even if when you're looking at a UMAP, there might be colors you want to adjust. So what you can do is click on that group that you want to adjust, and you can change the color as well for that specific cell type that you're trying to adjust that for. So that's where you're going to be able to select that individual sort of cell type, and you can change the color scale for that. Other advanced chart setups will be also available soon when it comes to looking at the actual statistical data. For example, for boxplots, you might want to see the actual sort of outliers, or you might want to add other statistical information. So that's something that we'll be releasing as well very soon. So now another thing to also mention is how can we export these figures from BioVinci into a better resolution? So I think that was there was a lot of requests from different teams where if you're saving this analysis, how can I export it in a different format or in a different resolution that would be better for publication purposes? So this is where you're able to select what file format you're trying to export the figure in, what sort of resolution you need the figure in, and is there any color mode that you need to adjust as well. So that's something that you can add here so that you're able to export the preview of the figure, and you can then sort of use that for your own use. Okay. So this is where you can start working on both systems. So this was sort of the second part, is really how can we map out these different proportions and actually annotate either density scores or annotate enrichment scores from an entire gene set across different cell populations. So that's sort of the second part. Now, if we go into the third question for the session today, is how can we actually look at hybrid populations and whether they have functional shifts. So can we see specific gene sort of expression values, and can we see shifts across different sort of cell types that might be sort of hybrid or non-canonical specific cell types? So for that specific question, let's say we go back into our data set where we have these different conditions and we have different cell populations that we want to focus on. So let's say we want to isolate these specific non-canonical or non-hybrid populations, which are the MSN subtypes within neurodegeneration. So if you're trying to sort of map out these specific populations, you can click on the cell subtype. And what you're able to do is select select which of those populations would be the focus or the specific hybrid cells that you want to focus on. So let's say we want to look at this hybrid population and we want to look at these non-canonical sort of spiny neuron cells. So what I'm going to do is select the three specific cell populations, and I'm going to subcluster them or isolate them from the entire UMAP. So now that we have these populations only from the study, we can do a second subclustering in case if you want to only look at like Alzheimer's disease, only focusing on these populations, or if you just want to look at healthy donor samples within these cell types, you can also do a second subclustering so that you only look at those specific samples or those disease groups within these cell populations. Okay. So now if we want to look at, okay, is there any functional shifts that might give us some indication whether there's genes that are expressed like metabolically or that are driving specific differences within these cell populations? So if we go into one of our gene sets here that is looking at metabolic precursor genes or looking at sort of stress specific genes, we can search for the expression of these individual markers, and we can actually create a not a cell proportion, but we can create a bubble heat map. So what that's going to allow us to do is actually look at the proportion changes of the cells across these different populations, and also the level of expression. So one is the mean expression value that is showing you the level of expression within that cell type that is expressing that specific gene. And the coverage is the percentage. So the size of the actual sort of or the node is showing you that coverage expression. So how many percentage of cells are within that cell type that are expressing that gene. So that's other types of figures that you can also generate within BBrowserX Pro. And you can also change or split it by different conditions. So one way that we can look at it is, okay, is there any differences across these different condition groups? So I'm able to create these different plots as well. And can I actually configure it so that it's showing me the relative? So the scale mode is another sort of parameter that you can change when you're looking at expression of specific genes. So you can change it to either a relative sort of scale mode or an absolute scale mode. So that will be able to show you within that study, am I seeing these differences within these different populations? So you can include non-expressed genes as well if that's something that you need to add a layer in the analysis, or you can start exporting it into Vinci as well. So you'll see here, normally you're not able to see the plot clearly, like the whole entire sort of title for the cell type annotation. So when we export them into Vinci, you'll also be able to adjust the plots individually. So you're able to actually see the entire sort of title. So if we edit the plots, you can actually change either the height or the sort of width of that. And you can also change the height so that it allows you to be able to visualize the plot, or you can change the direction as well of the axis. So if you need to change the y-axis or the x-axis, or you want to collapse or add other information, you can do that as well for the plot. Okay. Okay, so I'm just going to sort of show it here. So now that we have this, we can see sort of the annotations, or we can actually change the annotation name as well. Okay. So I think for here, these are some of the main focuses that I wanted to show is really how are we able to generate these different tools? How can we search for very specific genes that might have functional shifts within specific cell populations? And can we then export them in a better resolution or adjust the plot so that it's clear, or we can add specific either color scale changes or actually change sort of the title or how we're able to visualize that data in the system. So again, there'll be a lot more, I guess, adjustments or parameters that you can add to the figure in the advanced settings, which are coming very soon. And these advanced settings will differ depending on the type of figure that you're generating in the system. So if it's specific to proportions, if it's specific to gene coverage, or if you're trying to look at cell intersection analysis, that's also another sort of plot that you can generate in the system, and we'll be sort of allowing you to export that very soon to look at these intersections across different cell populations. Okay. So if I go back into my slides, so some things that I want to mention is that we now give you the ability to just keep track of what roadmap features will be available in BioVinci. So if you go into the actual BioVinci platform, you'll be able to see here that we include sort of information on the roadmap, and that will give you an idea of its features that are sort of features that are quite important in your workflow analysis, or are there still features that are missing within the roadmap that would be very useful to add, especially when it comes to visualizing data or different statistical parameters that might be useful to integrate into these different figures. So for example, here we'll be adding very soon the basic statistical analysis. So really being able to not only generate like gene coverage, but actually being able to run further sort of statistical sort of ANOVA or other tests that you can sort of identify this is significant or not. So that's other sort of features that will be available soon, as well as how you manage color palettes across other figures in the system that are not yet have that exporting feature. And really, how can we share these different figures with other members of the team? So that's also something that we're planning to release very soon. Okay, so if there is any specific analysis that you would want to request from our team, feel free to either email the support email and just let us know that it's specific features that you would want to see in BioVinci or something that you would want to see in BBrowserX Pro so that you can export it into BioVinci. Okay. And if I go here, so some of the summaries that we were able to do or able to look at, looking at this specific case study relating to neurodegeneration, is really how can we look at different functional shifts within specific cell populations. So that would be through the AUCell enrichment score that again you can find when you're searching for particular gene targets. You can search for the full list. It can be even 50 genes, and you can then create an AUCell enrichment score distribution from the entire gene set. So that can give you an indication if that enrichment is only specific to a population within your data or to a specific disease group. The second part of the case study was really looking at how can we look at activated populations. So can we look at increased gene sets and can we see sort of abundance across specific sort of control groups or other disease groups. So that's through the density layer. So if you are planning to run that, it is now available for you when you're looking at an individual gene within the UMAP. You're able to now look at the density of that particular gene and look at the layer of that density as well. So that's a feature that you're able to run currently. Okay. And the third part of the case study was really looking at how can we create these different pathway enrichment scores and actually look at proportions. So in order to do that, make sure when you're running any AUCell enrichment sort of analysis, so let's say if we're looking at the enrichment of all these genes, when you're running AUCell enrichment, you need to make sure that you're saving the AUCell score. So for example, if I want to look at all the genes and I'm running the AUCell enrichment score, make sure that you either save the entire sort of threshold of that enrichment into your metadata first. You can add the title so that you know what AUCell score that's coming from or what gene set that's coming from. And once you save it, it'll add it into your metadata as a numerical sort of score input value where you can now create sort of a categorical value from that unit. So if you want to be able to create, let's say from this neuroinflammatory gene set, I want to only look at specific cells that are enriched at a higher threshold, you can select that threshold and create a new annotation. So that will create sort of a categorical variable so that you're able to look at proportion differences across different pathway gene sets. Okay. So that was sort of the session for today and some of the new features that we've been adding and some of the new releases within BioVinci for more data visualization editing of specific figures. So I hope that this is useful and that you're able to start using it within your own analysis or your own data. So I guess the next part, if there are any questions from the users in the team or the users who joined us today, let us know in the chat so that we're able to see those specific questions. And if there's not any questions, but you just want to ask something one-to-one that is specific to your data, you can schedule a one-to-one session as well. Okay. Okay, so I don't see any specific question at the moment from the chat. So I hope that this was useful. And again, we will be holding these specific webinar case studies every week. So there'll be one available for the Europe and Japan time in the next week. So we try to sort of distribute these specific sessions so many users from different sort of teams can join as well. So our next sort of the Eastern time webinar session will not be next week, but the coming week after that. So I hope to see everyone who joined today, and we'll have a new case study that might either be smart bulk or spatial data. Yes, Tian, I think you have a question. Okay. Or Kumar, you had a question. Okay. So yeah, if you want to ask a specific question for your specific data, you can send us that question specifically, and we can sort of support you on that if it's specific to your data. Okay. So thank you so much for everyone who's joined today. So I hope to see you in the next coming webinars. And again, if you have a specific case study request, like a specific platform or data set that you want us to use for the webinar to show you specific features, let us know if that's something that you need help on. So no, there's just to give a clarification on one of the attendees. So no, there's no limitation on the number of attendees. So if you have any other colleagues that want to join, please feel free to invite them. So there shouldn't be any issue with that. It might just be that we're just sort of turning on the sort of group, and that's why it might be hard to join. So please just let us know if there is any issues on that that you're facing, and we can help you figure that out. Okay. Okay, so thank you, everyone.